Thanks to Brilliant for supporting this episode of SciShow. Go to brilliant.org/scishow to check out their computational biology course. Hot springs and hydrothermal vents might not be the first places you would look to find ways to fight a pandemic, but they've given us some of the most important tools we have for identifying, tracking, and understanding pathogens. In fact, without them, the entire field of molecular biology would still be stuck in the 1960s, and not in the fun way. See, that is when researchers discovered a microbe in Yellowstone that allowed them to actually replicate DNA at will. This process, known as polymerase chain reaction, or PCR for short, was really a game changer for genetics, and we here at SciShow do not use that phrase lightly. The pathway to developing PCR began way back in the middle of the 20th century. Scientists had really just seen DNA for the first time, but already they were searching for ways to read these genetic blueprints. The problem was, sequencing DNA and its simpler cousin RNA required a lot of genetic material because it relied on cutting longer strings into smaller ones with enzymes that sliced at certain sequences and then back-calculating what the whole string looked like from those pieces. And simply put, it wasn't easy to obtain that volume of genetic material, especially for anything not microbial in nature. So they knew that being able to make copies of DNA would open the door to all kinds of research. And the potential to do that traces back to 1957, just a few years after the structure of DNA was first described, when researchers identified the first DNA polymerase. These enzymes build strands of DNA from nucleotides, the essential building blocks of nucleic acids. They take a single strand of DNA as a template and then string together a complementary strand. And all organisms have the blueprints for at least one of these enzymes written into their genetic code. That's how they make copies of their genome when their cells divide. Researchers soon discovered that they could isolate these enzymes from bacteria like E. coli, which meant, hypothetically, they could use them to replicate any DNA they wanted, except there's a small catch. Before the polymerase can start copying DNA, the tightly wound paired strands of DNA found in organisms like us have to be separated into single strands. That way, short sequences of single-stranded DNA, called primers, can bind to the open strand and tell the polymerase where to start and stop copying. In nature, this unwinding requires yet another enzyme, plus several other proteins too complicated to recreate in a tube. Luckily, DNA strands can be separated another way. Above 90 degrees Celsius, the bonds holding the strands together break apart. So you can heat the DNA up to get single strands, then cool things down a bit to let the primers bind, and so DNA polymerase can start making copies. After each heating and cooling cycle, you essentially double the amount of DNA. Today, each of these cycles takes about 5 minutes, so within a few hours you can go from a very small number of copies to millions of copies of a given DNA sequence. But back in the 60s, the whole process it took a lot longer than that. See, heat also permanently inactivates the DNA polymerases from E. coli. So in early DNA replication efforts, fresh polymerase had to be added each time a copy of DNA was made, making the process very slow and very expensive. That is where extreme microbes come into play. In 1966, scientists discovered a microbe living in the 70+ degree waters of hot springs in Yellowstone National Park. They named it Thermus aquaticus after its ability to thrive in the hot springs' high temperatures. And in 1976, researchers isolated one of its DNA polymerases. They called it Taq polymerase, or just Taq for short. And like the microbe itself, it could withstand the high temperatures needed for separating DNA strands. So you could throw it in with your sample, some primers, and nucleic acids, and then let a machine heat and cool everything over and over again to produce millions of copies of DNA. And that, in a nutshell, is polymerase chain reaction. Ever since researchers developed these methods in the mid-80s, PCR has allowed scientists to turn teeny tiny amounts of DNA into much larger ones in a matter of hours. For geneticists, it was like going from carbon copy paper to a Xerox machine. It meant they could figure out whose cells are on the end of a hair at a crime scene, or spot the genes of a virus hiding in someone's blood. In short, Taq polymerase revolutionized genetics, and it's still widely used for PCR today. But newer enzymes are coming into play too. See, Taq sometimes makes mistakes. It grabs the wrong nucleotide and attaches it instead. This is a problem for applications that need a high level of accuracy, like if you're trying to detect the small mutations that can happen to a virus over time. Doing that can help experts understand how the pathogen is moving around and changing. Luckily, researchers discovered Pyrococcus abyssi, a deep-sea microbe that lives in even more extreme conditions. Its enzymes are more resilient than Taq, but more importantly, they also proofread themselves while making copies, which is why they are 40 to 50 times more accurate. And they are now among the several specialized polymerases available to geneticists. And researchers keep going back to extreme environments because their microbes have all sorts of unique and useful molecular tricks up their sleeves. So who knows what else we will discover by studying these remarkably resourceful organisms. PCR didn't just provide new tools for studying viral outbreaks, of course. Suddenly, scientists could use genetic information to study all sorts of evolutionary and biological questions. And you can learn about all of that with the computational biology course from Brilliant. Brilliant offers dozens of interactive and engaging STEM courses to take your science, math, and engineering skills to the next level. And with their computational biology course, you can really dig deep into all the amazing things researchers can learn from sequencing genetic molecules. Plus, if you are one of the first 200 people to sign up for an annual premium subscription at brilliant.org/scishow, you will get 20% off. So, if you're interested, check it out.